this is a B4 video for the topic bioenergetics, looking at photosynthesis. And in this video we're going to be looking at the experiment where we compare the plant that's been in the light with the plant that's been in the dark in terms of the starch present in the plant. So let's rewind a little bit. We know that photosynthesis produces glucose. We know that photosynthesis requires light. And we also know that glucose is a small molecule, soluble, and it can be converted into the larger insoluble molecule that's starch for storage. So we've got this situation where we've got a plant in the light, plant in the dark, and we need to see which one has still got its store of starch. Here's the method. So we first of all put the leaves into boiling water, okay, and that helps to break down the cell wall. And then we put the boiling tube with the leaves in it into the hot water to keep it warm, and into the boiling tube we add ethanol. And what the ethanol does is it removes the chlorophyll from the leaf, so it's easier to see the colour change when we add the iodine solution. So let's have a look at the parts of the experiment. So we first of all left the leaves in the boiling water for 2 minutes. Okay, that breaks down the cell wall, allows us to access the contents of the cell. We used a Bunsen burner to heat up the water. We might have used a kettle because it's safer. Now it's safer because the ethanol that we use later is flammable. So if we've got the risk of Bunsen burners on and we're using the ethanol, and some of that ethanol was to spill on the Bunsen burner, then we've got a really flammable situation, a very dangerous situation. So we tend to use kettle water. That's sufficient for breaking down the cell walls, and then the ethanol is used to remove the chlorophyll from the leaf. And then later we add iodine solution to see if the leaf contains starch. And what we tend to see is that the leaf that's been from the plant that's been in the daylight has got plenty of starch, which makes sense. It's been photosynthesizing, it's produced some glucose, some of that glucose that it hasn't used straight away, it's converted to starch for storage. So the iodine in the leaf that's been in the light will change from orange to blue-black. Now the leaf that was in the dark, it hasn't photosynthesized, so it's needed to break down that starch reserve and use it as glucose. And we think of this as like a midnight feast. Okay, so the iodine solution, when you add it to the leaf that's been in the dark, does not change colour, it stays that orange colour. Now we can show this quite well on a variegated leaf as well. Okay, variegated leaves have sections of the leaf where there's no chlorophyll at all and they appear as white, usually around the edge of the leaf. And we can see that when we've done the starch test using the iodine here, we can see we've got blue-black on the green areas, okay, because there's starch there. And then on the variegated areas where there's no chlorophyll, the iodine solution has not changed colour. It stayed that orange colour because there's no starch in that area, because that part of the leaf has not photosynthesized. So this isn't comparing light and dark, this is just comparing presence of chlorophyll or absence of chlorophyll. Where there's chlorophyll, on a sunny day, you'll have the photosynthesis, therefore some of that glucose will be stored as starch and will change the iodine solution from orange to blue-black. And once again, you can see here the white areas, the iodine does not change colour, it stays orange because there's been no photosynthesis there because there's no chlorophyll. We can also see this in an exam question situation like this, where rather than putting the whole plant in the dark or in the light, part of the leaf is just covered up with some black paper. And we can see again, similar to the variegated leaf, if there's no light on this middle section, there will be no photosynthesis, the chlorophyll can't absorb the light, so therefore there's no starch there. And we can see that because the iodine hasn't changed colour. And then in the areas exposed to the light, top and bottom there, we get that blue-black colour change because the starch is present there. It's been made in terms of through photosynthesis as glucose, and then the glucose has been converted into that starch and stored, and we can see that colour change to blue-black. So let's look at some concluding questions. Explain how starch is formed in the leaves. Okay, so photosynthesis makes glucose. The plant converts some glucose to starch for storage. Identify what the iodine solution shows. The iodine is the solution we use to test for starch. So it starts off orange. If there is no starch, then the iodine solution will stay orange. If there is starch, we get a colour change to blue-black. And then describe the results of each plant and explain what this means. The plant in the light photosynthesized, produced glucose, converted some of that glucose to starch. When we tested it with iodine solution, we found that it went blue-black, which is a positive test for starch. The plant in the dark wasn't photosynthesizing because it wasn't absorbing any light. There was none. So it used some of that starch storage that it made earlier in the day, broke down that starch into glucose, used it. So the iodine solution has a negative test for starch because there's no starch present. It remains orange. Hope you found that useful. Thank you.